Hey everyone, so for this video, we're going to talk about phage infections. And remember, phage is also known as bacteriophage. And remember, phage, this is a type of virus that infects bacteria. And remember, phage are very specific. They only will infect bacteria, so they won't infect humans, animals, plants, anything else. So these two types of infections I'm going to talk about today are specific only to bacteria. This is not the same thing that happens in an animal virus. That will be a different video. So there's two types of phage infection. The first is called the lytic cycle, or a lytic phage would cause a lytic cycle. So here's what this looks like. So here we have a bacterial cell. Right now it's nice and happy. It has no clue what's going to happen. Here is the bacterial chromosome, the DNA of the bacteria. So now, step 2, we still have our happy little bacteria and its happy little DNA, and along comes the phage. So remember, phage are a type of complex virus. They have a complex shape. So this is complex. This round part up here, that's the capsid, which is the protein coat. And the capsid is going to protect that nucleic acid, which is inside of it. The phage has these little arm-like things, and these are called the tail fibers. And the tail fibers are going to attach to the bacterial cell. They're going to attach to a receptor on the cell and get ready to cause their infection. So now the phage is attached. What's going to happen next? We still have our bacteria, the phage is attached there, and we still have our DNA. Now the phage is going to insert its DNA into the bacterial cell. So the phage DNA is inserted, and actually the phage produce a little bit of lysozyme that kind of creates a little hole in the bacterial cell wall, and that's where the DNA can get in. Now this part on the outside, what's left of the phage, becomes inert. It's useless. There's nothing left to it. It's just protein. So that will eventually just get chopped up and it'll just go away. So that cannot go off and infect any more cells. It's done. Now's where the scary stuff starts to happen, and the bacteria is going to be very upset. So now that the phage DNA is in the cell, it's going to start taking over the bacteria cell's components, like its ribosomes, its DNA polymerase, etc. And so the virus is going to start making more copies of its nucleic acid, and then from there, it's going to make more copies of its capsid, more copies of its tail fibers. In this stage, the virus is replicating its parts. And the next stage, the virus is going to start to assemble its parts, so we call this stage assembly. Okay, so we're going to have new virions form inside of the bacteria, and this could be thousands of virions, of course. And of course, they're all going to have that DNA. And then as you can imagine, the last step, because this is the lytic cycle, the last step is the virions are going to release some lysozyme, which is going to pop the cell open, and the phage viruses can exit the cell and go off and infect some new bacteria. So this final stage is called release. Alright, and if you don't like my pictures because they look like a 5-year-old drew them, here is a pretty picture down here. Okay, remember, the phage attaches to the bacteria. It inserts its DNA into the bacterial cell. It starts synthesizing some new parts, which will eventually assemble. And then finally, the virions will be released from the bacteria. That is, again, the lytic cycle. Let's talk about the second phage type of infection. So the second infection is called the lysogenic cycle. Lysogenic cycles happen with temperate phages. So temperate phages can undergo a lysogenic cycle. So let's talk about how this cycle differs from the last one. So again, we have this happy little bacterial cell with its chromosome here. And once again, we're going to have a similar looking phage come in and attach to the bacteria. And here's its DNA inside the capsid. So far everything is the same. Then again, the phage is going to insert its DNA into the bacterial cell. Here's where the process changes from the lytic cycle. Instead of replicating and making more copies of the virus, instead the virus is going to insert its DNA into the bacteria's chromosome. Right here is where you see the difference. The phage inserts its DNA into the bacteria's chromosome. The phage uses a special enzyme called integrase. So integrase allows the phage to insert its DNA into the bacterial cell. Now what's going to happen? Anytime this cell undergoes binary fission, so anytime it replicates, all of its new baby bacterial cells are also going to have that phage DNA inside of them, inside of their chromosomes. So this is a permanent change, all right? All the daughter cells are also going to have that phage DNA. So this is where it becomes really important and really critical. Now, normally, this cell probably won't turn into a lytic cycle, but it could, okay? It has the potential to change to a lytic cycle. So, for example, if the cell is stressed because of UV light or chemical stress, what can happen is the DNA can reactivate, in a sense, and now the virus can start replicating itself again, okay, assembling, and then eventually, again, the cell can lyse open and release those viral particles from the cell. So again, something like UV light or stress could induce this change. When phage DNA over here in this picture, when you see phage DNA inside of a bacterial chromosome, this is called a prophage. So prophage is viral DNA inserted into the bacterial chromosome. So once again, pretty picture. A virus comes in, phage, and it's going to attach to the bacteria, and it's going to inject its nucleic acid into the cell. The nucleic acid could immediately start replicating and forming new virions, which will lyse the cell and go off and cause more infection. Or the phage DNA can insert itself right here into the bacterial chromosome, in which case we call it a prophage. It can hide out there for a long time, and any new daughter cells will also have the prophage DNA. Or it could switch and again become a lytic cycle, so we could go this route and go back, and again it could cause a lytic cycle. The last thing I want to talk about with this is this right here, the prophage. Okay, what can happen to the bacteria if it has this phage DNA inserted into it? And from a medical standpoint, why do we care? And here's why. A lot of times, phage DNA, the phage DNA that's inserted into the bacteria, might carry genes for toxin production. So it can carry genes for toxin production. So if the bacteria, such as these guys here, okay, start replicating, and they all can produce toxins based on this phage DNA that's inserted in them, that could have devastating effects on the host. And some examples of human bacteria that contain phage DNA that encode for toxins, and you guys have heard a lot of these, okay, Corynebacterium diphtheriae, which causes diphtheria, botulism, which we've talked about, caused by Clostridium botulinum, okay, E. coli, this is a very pathogenic strain of E. coli, O157H7, Vibrio cholera. So all of these examples in this chart produce toxins because of phage DNA, which is inserted in the bacteria.